the last time you cut yourself. Something happened that you probably did not think about. The bleeding slowed. Then it stopped. Not because you did anything, not because you pressed hard enough or long enough, because something inside you, something ancient and automatic and running without your awareness, sealed the breach and kept the river moving. You watched 600 million years of evolutionary engineering work in real time, and you thought nothing of it. There is a rule built into the physics of this planet that shapes every living thing on it. It is not a rule anyone wrote down. It was there before anything capable of noticing it existed. It governs the movement of oxygen through biological tissue, and it applies with the same indifference to a bacterium drifting in a warm ocean 3 billion years ago, and to the cells lining the inside of your lungs right now. Oxygen moves through living tissue by drifting. It spreads from where it is concentrated toward where it is scarce. Slowly, passively, without being pushed or carried or guided. This process works beautifully across very short distances. Across distances measured in hundredths of a millimeter. It is fast enough to keep a cell alive. Across distances slightly larger than that, it is not. The number that matters is roughly 200 micrometers, about the width of 2 human hairs pressed together. Beyond that distance, oxygen runs out before it arrives. The cell waiting on the other side does not receive what it needs. It does. 200 micrometers. That is the wall. For most of the history of life on this planet, everything that existed stayed on the right side of that wall. The ocean was full of living things, extraordinarily diverse and extraordinarily successful, and nearly all of them were invisible to the naked eye. Flat, thin, small, not because life lacked the capacity for something more, because the wall was real, and the consequences of crossing it were immediate and final. Then life started building bodies with depth, bodies with layers of cells stacked on top of each other, with tissues pressed together, with organs tucked inside other structures, bodies that were, by the brutal arithmetic of diffusion, too thick to survive. The wall said no. What happened next is what this story is about. Life did not break that rule. Nothing in the history of biology has ever broken a law of physics. What life did instead was find a way to make the rule irrelevant. It built something inside itself that carried what diffusion could not. Something that moved. Something under pressure. Something that, in your veins, right now, is making it possible for you to sit here and follow a thought from one sentence to the next. The ocean outside the body is where life began. The ocean inside is what let life leave. That second ocean is running through you right now. Without your awareness, without your instruction, without your permission. It has been running without stopping since before you took your first breath. It is the answer to a problem that took evolution hundreds of millions of years to solve. And it is so woven into what you are that you have never had to think about it once. You are about to think about it now. Before there was blood, there was a different answer to the same problem. And for a very long time, it worked perfectly. Picture the ocean roughly 600 million years ago, not the ocean as we know it today, with its fish and its whales and its coral reefs. An older ocean, quieter, stranger, an ocean that had already been home to living things for more than 3 billion years, in which the dominant form of complexity was still measured in micrometers rather than centimeters. This ocean was full, extraordinarily full. Bacterial mats covered the seafloor in layered sheets. Single-celled organisms of remarkable chemical sophistication drifted through the water. Each one, a complete living system, contained within a membrane thinner than a soap bubble. Colonies of cells had already begun experimenting with staying together, with dividing labor, with the earliest forms of multicellular existence. And every single one of these organisms shared the same relationship with oxygen. It arrived by drifting. For a single cell floating in oxygenated water, this is not a problem. The cell is so small that the distance between its outermost membrane and its innermost chemistry is trivially short. Oxygen arrives almost instantly. Waste leaves almost instantly. The cell lives in continuous contact with its environment. And the physics of diffusion is not an obstacle at this scale. It is a gift. The earliest multicellular organisms understood this. In the way that evolution understands anything through survival, the forms that worked were the forms that persisted. And the forms that worked were overwhelmingly the forms that kept every cell close to the water outside. Flat, thin, spread out, not because evolution was being cautious, but because the physics of oxygen diffusion made any other shape a sentence. Look at a sponge, a living one, anchored to a rock in shallow water, drawing a current through its body. A sponge has no heart, no blood, no circulatory system of any kind. What it has instead is a body plan of remarkable cleverness, a porous structure threaded through with channels that bring seawater into contact with nearly every cell in the animal. The sponge does not send oxygen to its cells. It builds itself so that the water carrying oxygen never has to travel far to reach them. A jellyfish can be several feet across, but look at the geometry carefully. A jellyfish is more than 90% water by mass, a thin layer of living tissue sandwiching an inert, gelatinous core. The cells that matter are never far from the surface. The jellyfish is large in the way that a soap bubble is large, a thin skin enclosing mostly emptiness. It is not cheating the diffusion limit. It is respecting it while growing as large as that respect allows. These are not failures of evolution. They are not organisms waiting to become something more sophisticated. They are exquisitely refined solutions to a genuine physical problem. Solutions that have persisted for hundreds of millions of years, because the problem they solve has not changed. Diffusion still works across short distances. These animals are still short-distance animals. The solution still fits. But a solution that works under one set of conditions does not automatically work under different conditions. And somewhere in the late Precambrian, conditions began to change. The physics did not change. The 200 micrometer rule was as absolute as it had always been. What changed was what life was trying to build. The fossil record of the Ediacaran period, roughly 635 to 540 million years ago, shows something that had not appeared before: bodies with genuine depth, bodies with tissues stacked in layers, with distinct regions performing different functions, with the beginnings of what would become organs, bodies that were, in purely physical terms, trying to do something that diffusion alone could not support. Some of these early experiments simply did not last. The strange, quilted and frond-like organisms that populated the seafloor during this period left behind fossils but very few descendants. Some may have been thin enough throughout their structures that diffusion still worked despite their apparent size. Others may have run into the wall and not survived the encounter. But some lineages found something new. They found a way past the wall that did not require staying thin. It was not elegant at first. It was the first rough draft of an idea that evolution would spend the next 600 million years refining into something extraordinary. A flow of fluid inside the body itself, carrying what diffusion could not, reaching cells that were too deep and too far from the surface to receive what they needed any other way. Life had not invented blood yet. But it had discovered that moving liquid was the answer. Here is the part worth pausing on. Right now, in your own body, there are still cells that face the same wall those Ediacaran creatures faced. The cells at the center of your eye's lens have no blood supply at all. They survive on diffusion alone, which is why they cannot divide, cannot repair themselves, and why the proteins inside them cloud over a lifetime into what we call cataracts. The wall that shaped 600 million years of evolution is still shaping your body today, in the places where the river never reached. The question that life needed to answer next was not whether to move fluid. That much was clear. The question was what to put in it. 540 million years ago, the ocean started building things it had never built before. Not gradually, not in the slow, barely perceptible way that most evolutionary change happens. By geological standards, what occurred at the beginning of the Cambrian period was something close to an explosion, within a span of time that sounds long by human measure, but registers as a single sharp moment in the 4 billion year record of life. The ocean filled with animals of a complexity that had no precedent anywhere in the fossil record. Eyes that could resolve detail in dim water, mouths that could open and close with force, shells of calcium and chitin hard enough to deflect attack, muscles capable of generating real speed, nervous systems capable of coordinating that speed in response to what the eyes were seeing, digestive systems with distinct regions performing different chemical tasks, bodies with a front end and a back end, a top and a bottom, an inside that was genuinely inside and protected from the world outside. Real bodies. And every single one of those bodies had the same propulsion written into it. Depth. A body with muscles has layers of muscle tissue that no surface can reach. A body with a digestive tract has cells lining that tract surrounded on all sides by other tissue. A body with a nervous system has neurons clustered together, insulated from the outside world. These are not incidental features of complex animal design. They are what complex animal design is. Complexity requires cells that are committed to one function and isolated from the environment. Isolated from the environment means isolated from the oxygen in the water outside. The 200 micrometer rule had not changed, but the bodies life was building were now routinely demanding more than that rule allowed. Consider Anomalocaris, one of the most studied predators of the Cambrian Ocean. It reached lengths of perhaps half a meter to a full meter, a genuine giant by the standards of its time. It had compound eyes of real sophistication, capable of resolving detail in dim Cambrian water. It had grasping appendages at its front end, flexible and segmented, built for catching prey. Its mouth was a circular structure of overlapping plates that could close around a victim with significant force. Everything about Anomalocaris, its size, its apparent speed, its predatory apparatus, suggests a level of metabolic activity and muscular coordination that simple diffusion could not have supported. Its size alone is a kind of argument. Bodies at that scale, with the kind of active lifestyle the fossil evidence suggests, strongly imply that something was moving oxygen through its tissues beyond the passive drift of diffusion. The exact nature of its internal fluid transport is not preserved in the fossils we have, but the existence of the animal is evidence that the problem had been solved, at least partially, at least well enough. Anomalocaris was not alone. The Cambrian Ocean was filling with animals of comparable complexity. Animals that were building the first arms race in the history of life. Predators developing better eyes and faster appendages. Prey developing harder shells, and more effective escape behaviors. Every escalation in that arms race demanded more energy. More energy demanded more oxygen. More oxygen demanded a better system for getting it to where it was needed. And fast. The diffusion-based solution that had worked for billions of years was not failing. It was being outgrown. The animals that could solve the delivery problem were the animals that could compete, the ones that could not left no descendants complex enough to tell. Here is what makes this moment so strange to think about. The problem that drove the invention of circulatory systems was not imposed from outside. It was a problem that life created for itself by wanting to become more complex. The wall that diffusion built had been the ceiling of animal complexity for hundreds of millions of years. The Cambrian explosion was the moment life started pushing against that ceiling hard enough that it had to find a way through. And the way through was not a single invention. Evolution does not hold a design competition and select a winner. What happened instead was that multiple lineages of animals, facing the same physical problem, arrived at variations of the same solution through entirely separate paths. Move the fluid, create a circuit, pump it. In some lineages, the fluid moved through open spaces within the body cavity, washing over organs directly. In others, it moved through increasingly defined channels that would eventually become true vessels. In some, the pumping was done by the rhythmic contraction of the body wall itself. In others, a dedicated region of tissue began specializing in nothing but contraction, generation after generation, until it was no longer recognizable as anything other than what it had become, a heart. None of this happened quickly. The transition from a body that relied on diffusion to a body with a functioning circulatory system was not a moment, but a direction that different lineages traveled at different speeds and arrived at in different forms. But the direction was the same, because the problem was the same. Distance had become deadly. There is something in that sentence worth making personal. Think about the last time you held your breath underwater, or ran hard enough that your chest burned, or felt your heart hammer after a sudden shock. What you were feeling was your circulatory system responding to a sudden mismatch between oxygen supply and oxygen demand. Your heart rate climbed. Your blood vessels adjusted. The whole system shifted in real time to close the gap. That response, so automatic, you barely notice it, is the solution that the Cambrian Ocean spent tens of millions of years working out. Every time your body responds to effort, it is running code written in the fossil record of animals that had no choice but to solve the problem or disappear. Life stopped waiting for oxygen to arrive on its own. It went to get it. The question was, what kind of fluid could carry enough oxygen to make the journey worth making? And the answer to that question turned out to be far stranger and far older than anyone would have guessed. Blood is red. This is one of the most basic things a person learns about their own body. So early and so completely that it stops feeling like information and starts feeling like a fact of nature. Blood is red the way the sky is blue. It simply is. Except that it is not always. Not for most of the animals that have ever lived. The fluid running through the body of a horseshoe crab is pale blue. Not blue in the way that veins sometimes look blue through pale skin, which is an optical trick. Genuinely blue. The way copper-based paint is blue. Because the molecule doing the work of carrying oxygen in horseshoe crab blood contains copper rather than iron. When that molecule picks up oxygen, the copper changes state and the fluid turns blue. When it releases the oxygen, the color fades to near colorless. The horseshoe crab has been on this planet for roughly 450 million years. It survived the extinction at the end of the Permian period that killed more than 90% of marine species. It survived the asteroid impact 66 million years ago that ended the age of the dinosaurs. Its blood is blue. It has been blue for the entire span of that survival, and there is no meaningful sense in which blue blood is inferior to red blood. It is a different solution to the same problem, one that worked well enough to carry a lineage through nearly 500 million years of catastrophe. The fluid moving through the body of an octopus is also copper-based and also blue. Octopuses have 3 hearts, 2 of which pump blood specifically to their gills, and 1 of which pumps it to the rest of the body. Their problem-solving ability is extraordinary. Their ability to change color and texture in under a second is unmatched anywhere in the animal kingdom. And their blood is not the color of anything we typically associate with the word. Some marine worms carry a molecule called hemerythrin, which turns violet-pink when it binds oxygen and colorless when it releases it. Certain sea cucumbers use the same molecule. Antarctic icefish have lost their hemoglobin almost entirely over the course of evolution, relying instead on an unusually high volume of blood and unusually large hearts to move enough oxygen through their near-freezing bodies. Their blood is nearly transparent. The reason all of this matters is not that it is surprising, though it is. The reason it matters is what it tells us about how evolution actually works when it is solving a hard problem. Evolution did not invent blood once. It invented the idea of blood many times, in many forms, using different chemistry, different architecture, and different colors, because the problem it was solving was not make blood, but move oxygen and nutrients through a body too large for diffusion. That problem has a family of solutions. Different lineages found different members of that family, and all of them worked well enough to produce animals that have survived for hundreds of millions of years. The 2 most common oxygen-carrying molecules in animal blood are hemoglobin and hemocyanin. They share a function, but almost nothing else. Hemoglobin uses iron as the atom that grabs and releases oxygen. It is packed inside red blood cells and moves through a closed circulatory system under pressure. Hemocyanin uses copper, is dissolved freely in the blood fluid rather than contained in cells, and typically moves through an open circulatory system where the fluid bathes organs directly rather than flowing through enclosed vessels. Each approach has its own strengths and its own limits. Hemocyanin works remarkably well in cold water and at low oxygen concentrations. The horseshoe crab, the octopus, the lobster, the snail, all of them carrying their copper-based fluid through open or semi-open systems, have been extraordinarily successful in the environments where their chemistry is well matched to local conditions. The system is simpler. It requires less specialized architecture, and it has kept those lineages alive through extinctions that destroyed the vast majority of complex life on this planet. What it cannot do easily is generate high pressure, an open circulatory system, where the fluid fills body cavities and washes over organs rather than moving through enclosed vessels. Cannot be pressurized the way a closed system can. Pressure requires containment. Without containment, there is no pressure. Without pressure, the fluid moves slowly, and the maximum rate at which oxygen can reach active tissue is limited in ways that become significant when the body is large and the demands on it are intense. For a clam sitting on the seafloor filtering water through its gills, that limit is not a problem. The clam is not in a hurry. Its muscles are not firing at high frequency. The open system is well matched to the life it needs to live. For an animal that runs, or chases, or flies, or maintains a constant body temperature independent of the environment, the limit becomes a ceiling. And here is where the closed circulatory system, the one running in your body right now, represents something genuinely new. A closed system contains blood within vessels at all times. The fluid never leaves the circuit to fill body cavities. It moves through arteries and capillaries and veins in a sealed network, under pressure generated by a dedicated pump. Because the system is sealed, the pressure can be maintained. Because the pressure can be maintained, delivery can be fast. Because delivery can be fast, the system can respond to sudden changes in demand, flooding active muscle with oxygen-carrying blood within seconds of the demand beginning. The capillaries, the smallest vessels in a closed circulatory system, are narrow enough that red blood cells must pass through them in single file. This narrowness is not a flaw. It is the solution to the last step of the oxygen problem. At that scale, the diffusion distance between the blood inside the capillary and the cell just outside it is small enough that diffusion can cover the final gap. The circulatory system carries oxygen to within 200 micrometers of where it needs to go. Diffusion covers the last step. The wall is not broken. It is approached from the other side. And oxygen was never the only thing the river carried. From very early in the history of closed circulation, blood moved fuel and waste and heat and chemical signals, hormones and proteins and immune cells. An entire logistics system compressed into a fluid and driven by a pump. The oxygen story is the most dramatic part. It is not the whole story. Every time you feel a rush of adrenaline before something important, or notice that your skin flushes when you are embarrassed, or feel your hands go cold when you are frightened and your core stays warm, you are feeling the circulatory system doing things that have nothing to do with oxygen. You are feeling 600 million years of logistics, routing resources toward where the body needs them most. Different lineages arrived at different versions of the closed system through entirely independent paths. The ancestor of vertebrates found one path. The ancestor of annelid worms found another. The ancestor of cephalopods, the group that includes octopuses and squids, found yet another. All of them navigated toward the same destination by different routes, all driven by the same pressure bodies that needed more delivered faster than their predecessors had ever required. The red river running in your veins is one answer to a question that life has been asking for 600 million years. It is not the only answer, but it turned out to be, for reasons that have everything to do with a single atom sitting at the center of a molecule that evolution spent hundreds of millions of years perfecting. The most powerful answer yet found for large, active, high-demand bodies. That atom is iron. And the story of how it ended up in your blood begins not on this planet, but in the core of a star that died before the Earth existed. Every atom of iron in your blood is older than the Earth. Not older in the way that rocks are old, or oceans are old, or even the planet itself is old. Older in a way that requires stepping outside the solar system entirely. The iron sitting at the center of your hemoglobin molecules was forged in the collapsing core of a massive star, in a process that required temperatures of billions of degrees and pressures that exist nowhere on the surface of any planet. It was released into space when that star exploded, scattered across light years of empty void, drifted for millions of years through the cold dark between stars, and eventually found its way into the cloud of gas and dust that gravity was slowly assembling into what would become our solar system. The Earth formed around that iron. The oceans formed on top of the Earth. And the first living chemistry that would eventually become animal life formed in those oceans. In a world where iron was one of the most chemically available metals in the water, This matters because the question of why hemoglobin uses iron is not arbitrary. Iron had a specific combination of properties that made it especially powerful in the lineage that would become vertebrates. And that combination is the reason a molecule built around iron became the dominant oxygen carrier in vertebrate blood, rather than a molecule built around something else. Iron can hold oxygen without destroying it. This sounds simple. It is not. Oxygen is one of the most chemically aggressive elements in the periodic table. It reacts with almost everything it encounters. It rusts iron left exposed to air. It breaks down organic molecules. It damages DNA. It attacks the lipids that make up cell membranes. The same chemical reactivity that makes oxygen so valuable as an energy source. Its eagerness to accept electrons from other molecules makes it dangerous to the very structures that depend on it. The challenge that life faced in building a molecule to carry oxygen through blood was not just binding oxygen. Any number of metal atoms can bind oxygen. The challenge was binding it loosely enough to release it on demand. In the right place, at the right time, without allowing it to cause damage along the way. Iron in hemoglobin does this through an arrangement of extraordinary precision. The iron atom sits at the center of a flat, ring-shaped organic molecule called heme. 4 heme groups are nestled within a single hemoglobin protein, each one cradled by the folded arms of the protein in a way that controls exactly how tightly the iron holds its oxygen. The protein does not just provide a container. It tunes the chemistry. It adjusts the electronic environment around the iron atom so that oxygen binds with exactly the right strength, firm enough to pick it up in the lungs where oxygen is abundant, loose enough to release it in the muscles and organs where oxygen is scarce and where the slight shift in local chemistry is enough to tip the balance and let the oxygen go. This is a machine in the most literal sense, one whose function depends on the precise 3-dimensional arrangement of thousands of atoms working in coordination. And evolution built it through the same process. It builds everything: variation, selection, and time. A great deal of time. But here is where the story gets complicated. Because iron that is not inside hemoglobin, iron floating freely in the blood or inside a cell, is not a gift. It is a threat. Free iron catalyzes a reaction in which hydrogen peroxide, a normal byproduct of cell metabolism, is converted into one of the most destructive molecules in biology. These hydroxyl radicals attack DNA and break the strands. They tear apart proteins and destroy their function. They strip electrons from the fatty acids in cell membranes and trigger a chain reaction of damage that spreads like fire through dry wood. A cell with significant free iron inside it is a cell being quietly taken apart by the very chemistry it depends on to survive. Every system in the vertebrate body involved in handling iron is built around one overriding imperative: keep the iron contained. Hemoglobin locks iron inside the heme pocket where its chemistry is controlled. Red blood cells keep hemoglobin enclosed within a membrane, preventing it from leaking into the blood plasma where it could cause damage. Proteins called transferrin escort iron through the bloodstream between cells that need it. Ferritin molecules inside cells store iron in a safe, sequestered form, releasing it only when needed and only in amounts that can be immediately put to use. The entire iron handling system of the vertebrate body is an elaborate set of containment protocols built around a metal that is simultaneously essential and dangerous. Too little iron and hemoglobin cannot be made, and cells starve for oxygen. Too much free iron and cells begin destroying themselves from the inside. The balance is maintained through layers of regulation, so finely tuned that even small disruptions can throw the whole system into dysfunction. Hemochromatosis, a condition in which the body absorbs too much iron and cannot regulate its storage properly. causes iron to accumulate in organs, including the liver, the heart, and the pancreas, where the free metal gradually damages tissue over years and decades. Iron poisoning from acute overdose can be fatal within hours. The toxicity is not a side effect. It is the fundamental nature of the element that life chose to carry its oxygen. Evolution did not choose iron because it was safe. It chose iron because it worked, and then spent hundreds of millions of years building the containment systems necessary to make working with iron survivable. There is something worth sitting with in that sequence. The most important oxygen-carrying molecule in vertebrate biology is built around a metal that requires an entire additional layer of biological infrastructure to prevent it from poisoning the organism that depends on it. The gift came with a price. The price was paid not by rejecting iron, but by developing, layer by layer, the machinery needed to keep it under control. And the payoff was enormous. Oxygen dissolves in water only sparingly. Blood, plasma, the liquid portion of blood, can carry only a small amount of dissolved oxygen on its own. An animal relying on dissolved oxygen in plasma alone would need an impossibly large volume of blood moving at an impossibly high rate to supply an active body. Hemoglobin changes the equation entirely. By binding oxygen reversibly to iron, hemoglobin increases the oxygen-carrying capacity of blood by roughly 60 to 70 times compared to plasma alone. The same volume of blood, with the same pump, delivers dramatically more oxygen per circuit. In vertebrates, this is what made the closed circulatory system far more powerful than plasma alone could ever be. The pump and the pipes matter, but what flows through them matters just as much. Think about what that means in your own body right now. Every breath you take fills your lungs with oxygen that will spend the next few seconds bound to iron atoms that were made before the Earth existed, carried in a molecule that evolution spent hundreds of millions of years perfecting, moving through a system of vessels that is itself one of the most consequential pieces of engineering in the history of life. The iron in your blood is not a coincidence of chemistry. It is a solved problem, sitting at the center of a molecule that required the deaths of stars to produce and hundreds of millions of years of evolution to learn to use safely. The dangerous gift turned out to be worth accepting. But to deliver it where it needed to go, life needed something it did not yet have. It needed pressure. A river with no banks is not a river. It is a flood. A flood moves. It carries things. In a technical sense, it transports water from one place to another. But it cannot be directed. It cannot be concentrated. It cannot be made to flow faster in one channel and slower in another in response to where the need is greatest. A flood is undifferentiated movement. And undifferentiated movement, when the goal is delivering oxygen to specific tissues at specific rates in response to what the body is doing at any given second, is nearly useless. The open circulatory systems of insects and crustaceans and mollusks are not floods. They are more controlled than that. But they share with floods the fundamental limitation of operating at low pressure in an unenclosed space. The hemolymph of a grasshopper, the pale fluid that serves the role blood serves in vertebrates, moves through the insect body, driven partly by a simple tubular heart and partly by the movements of the body itself. It works. The grasshopper is a successful animal by any measure that matters in evolution. But a grasshopper cannot run a marathon. Its circulatory system cannot sustain the kind of prolonged, high-intensity oxygen delivery that a large vertebrate body with a high metabolic rate demands. You cannot push a fluid faster through an open space than the space itself allows. and open spaces let whatever pressure the fluid generates dissipate in all directions at once. A closed system changes this completely. When blood is contained within vessels, the pressure generated by the pump is not lost to the surrounding body cavity. It is transmitted through the fluid to every point in the network. The same force that pushes blood out of the heart pushes it through the largest arteries and on through the branching smaller vessels, all the way to the capillaries at the farthest reaches of the body. The pressure does not dissipate until the blood has done its job and returned to be pumped again. This is why closed circulatory systems made a qualitatively different kind of animal possible. It is not just that they deliver more oxygen per unit time, though they do. It is that they can respond. They can increase output when demand rises and reduce it when demand falls. They can redirect flow from one region to another as the body's needs shift. They can maintain delivery at a consistent rate even as the animal moves, encounters stress, or suddenly needs to run. The heart is the machine that makes this possible. And the heart, in its vertebrate form, did not appear suddenly. It emerged the way all complex biological structures emerge gradually, through a series of modifications to structures that already existed. Each change selected because it works slightly better than what came before. The earliest pump-like structures in animal evolution were simply regions of a fluid-filled tube that contracted rhythmically, nudging fluid in one direction slightly more effectively than body movement alone. In annelid worms, the earthworm being the most familiar example, there are muscular vessels near the front of the body that contract in sequence to push blood forward through a closed circulatory system of real sophistication. They share with the vertebrate heart a single thing: generate pressure, maintain flow, keep the circuit running. In the vertebrate lineage, the heart evolved from a simple two-chambered structure in early fish to the four-chambered heart that birds and mammals carry today. These were not steps on a ladder toward the human heart. They were different solutions to different circulatory demands, each suited to the metabolic reality of the animal carrying it. But together, they reveal a consistent direction toward greater separation of oxygenated and deoxygenated blood, toward higher pressure, toward more precise control of where the blood goes and how fast. The four-chambered heart that appeared in the ancestors of birds and mammals solved the separation problem by running two completely independent circuits through the same pump. The right side receives oxygen-depleted blood from the body and sends it to the lungs. The left side receives oxygen-rich blood from the lungs and sends it to the body. The two streams never mix. Blood, leaving the left side for the body, carries the maximum possible oxygen concentration, because it has just come directly from the lungs without being diluted by returning blood from the body circuit. This separation is not a minor refinement. It is the architectural prerequisite for warm-blooded metabolism, for large and energy-hungry brains, for sustained high-intensity physical activity. Every bird that flies, and every mammal that runs, and every human being who has ever had a thought that required sustained concentration is doing so on the basis of a circulatory architecture that keeps oxygenated and deoxygenated blood separated by a wall of muscle in the center of the chest. The pressure that system generates is remarkable in its own right. The human heart pushes blood out of the left ventricle at a pressure high enough to drive it through nearly 60,000 miles of blood vessels, from the largest arteries down to capillaries narrower than a single red blood cell, and back again, in under a minute. It does this roughly 100,000 times per day. It does this every day of a human life without rest, without pause. The vessels themselves are not passive pipes. Arteries have muscular walls that can contract or relax to adjust their diameter and therefore the resistance they offer to blood flow. This is how the body redistributes blood in response to changing demand. When muscles need more oxygen during exercise, the arteries supplying them dilate. When the digestive system needs more blood after a meal, the vessels supplying it open wider. When you are frightened and your body needs everything directed toward muscle and away from everything else, the vessels do that too, automatically, before you have consciously decided what to do next. The heart does not need to know where the blood is going. The vessels route it. And at the far end of the arterial system, in the capillaries, the closed system delivers its final answer to the problem that started this whole story. The capillaries are so narrow, and their walls so thin, that oxygen can move from the blood inside them to the cells just outside across a distance, well within the range where diffusion still works. The entire infrastructure of heart and arteries and veins exists, ultimately, to bring oxygen to within a few cell widths of where it needs to go. And then diffusion. The ancient, passive, slow process that has been the foundation of oxygen transport since life began, covers the last step. The wall still stands. It has never been breached. What the closed circulatory system built was a delivery system that brings oxygen to the foot of that wall, consistently, reliably, under pressure, at a rate that adjusts in real time to whatever the body is doing. That was enough. That was more than enough, because with that delivery system in place, something became possible that had never been possible before in the history of life on this planet. Feel your pulse right now if you want to. Press 2 fingers against the inside of your wrist and find it. That rhythm, so familiar you have stopped noticing it, is the sound of a pump that evolution spent hundreds of millions of years building toward. Every beat is pressure entering the circuit. Every beat is oxygen moving toward tissue that cannot wait. Every beat is the answer to a problem that killed countless lineages of animals that could not solve it. And what that pressure made possible next is the part of this story that most people never connect to the blood running through them. It made possible speed. By roughly 450 million years ago, the oceans were increasingly shaped by animals capable of something that had not existed before in the history of life. Active pursuit, not ambush, not filter feeding, not waiting for food to drift within reach of a stationary body, closing the distance between itself and another animal through sustained, directed, high-speed movement, maintaining that speed long enough to catch what it was chasing, and then doing it again. This sounds ordinary now because we live in a world full of animals that chase and flee, a world where pursuit and escape are so common that we take them entirely for granted. But they required something that the earliest animal bodies did not have. They required the ability to sustain high oxygen delivery to active muscle, not for a few seconds, but for long enough to matter, and to do it on demand, in the first moments of a chase, before there was any time for the body to slowly ramp up its systems. They required a circulatory system capable of responding fast. Every chase in the history of animals has been at a level invisible to any observer, a contest between delivery systems. The predator wins if it can sustain oxygen delivery to its muscles longer and at higher rates than the prey can sustain escape. The prey survives if it can outlast the predator's cardiovascular capacity. Muscle strength matters. Limb geometry matters. But underneath all of it, setting the ceiling on what muscle can do and how long it can do it, is blood. The fish that appeared in the Cambrian and diversified through the Ordovician and Silurian periods were the first vertebrates. And they brought with them the closed circulatory system and the hemoglobin-packed red blood cells that the vertebrate lineage would continue to refine for the next 400 million years. They were not the fastest animals in those ancient oceans by the standards of what would come later, but they were something genuinely new, animals capable of sustained activity driven by a circulatory system that could maintain oxygen delivery through effort rather than just at rest. The ocean rewarded this. Think about what sustained cardiovascular capacity actually means in practice for an animal trying to survive. The moment a threat appears, or the moment prey is spotted, the heart can accelerate from its resting rate toward its maximum within seconds. Blood carrying oxygen can be redirected from organs not immediately essential, the digestive system, the skin, toward the muscles that need it most. Almost instantaneously. The muscles can sustain near maximum contraction, not for a single burst, but for long enough to close a gap, or to open one. The bluefin tuna is one of the most revealing examples of what closed circulation under sustained selective pressure eventually produces. A bluefin tuna can sustain swimming speeds that exhaust most fish within seconds. It does this through a combination of features, several of which are directly related to its circulatory system. The tuna has a countercurrent heat exchange system built into the blood vessels supplying its swimming muscles. Warm blood, leaving the core of the body, flows in vessels that run alongside cooler blood returning from the gills. And the two streams exchange heat across their walls without mixing. The result is that the tuna's core muscle temperature is significantly warmer than the surrounding water, sometimes by 10 degrees Celsius or more. Warm muscle contracts faster and more efficiently than cold muscle. The tuna is running a targeted heating system for its most critical locomotor tissue, powered entirely by the architecture of its circulatory system. This is not a coincidence of anatomy. It is a refinement built on top of a refinement built on top of a refinement, each layer improving the rate and efficiency of oxygen delivery to the tissue that needs it most, until the result is an animal capable of crossing entire ocean basins at speeds that seem improbable for a creature of its size. The peregrine falcon makes the same point from the air. When a peregrine stoops on prey, folding its wings and dropping in a controlled dive, it reaches speeds approaching 350 kilometers per hour. The forces on its body during that dive and during the pull-out at the bottom are enormous. Its heart, beating at rates that can exceed several hundred times per minute during intense activity, pushes blood to its flight muscles and to its eyes, which must track and adjust throughout the dive with precision that no open circulatory system could support. Insects solved flight through a different oxygen delivery strategy, one brilliantly suited to their size and metabolic demands. But that strategy does not scale into the kind of high-pressure vertebrate performance that sends a falcon through the air at 350 kilometers per hour. The cheetah runs at over 110 kilometers per hour. But what is more impressive than its top speed is what happens in its cardiovascular system in the seconds before it begins to run. When a cheetah identifies prey and prepares to chase, its heart rate rises sharply before the sprint begins. The circulatory system is not responding to the demand of running. It is anticipating it, ramping up delivery before the muscles have even started contracting, so that oxygen is already arriving at near-peak rates the moment the first stride hits the ground. The gazelle running from the cheetah has the same capacity. Its heart and circulatory system can sustain speeds and directional changes that would collapse most animals within seconds. The extraordinary athleticism of African ungulates, their ability to maintain near maximum speed through sharp turns and sudden stops and abrupt accelerations, is as much a circulatory story as a muscular one. The blood arriving at their leg muscles during a chase is under high pressure, carrying maximum oxygen redirected away from every non-essential system in the body, delivered by a heart shaped by the same predation pressure that shaped the cheetahs. Both animals are running on 600 million years of cardiovascular evolution. The outcome of any individual chase depends partly on which evolutionary lineage refined its delivery system more effectively for this particular contest, in this particular terrain, on this particular day. Human beings sit in an unusual place in this story. We are not fast by the standards of the animals we share the planet with. A fit horse outruns any human over any distance shorter than perhaps 80 kilometers. A cheetah would cover the length of a 100-meter sprint before most humans have reached the halfway mark. By the metrics of raw speed that dominate most predator-prey interactions, we are mediocre, but we can run for hours. The human ability to sustain moderate speed running over very long distances, combined with our exceptional ability to dissipate heat through sweating, may have supported a hunting approach that some researchers call persistence hunting, in which early human ancestors followed large prey animals at a steady pace for hours until the animal, unable to pant and run simultaneously, and therefore unable to cool itself adequately, became vulnerable, whether or not this was a primary strategy. The physiology that would support it is real. Human beings have cardiovascular and thermoregulatory systems unusually well suited to sustained effort, and the blood supply to the skin plays a central role in both. When you exercise hard enough that your face flushes and your skin feels hot, you are watching the circulatory system route warm blood toward the surface to shed heat through the skin. When sweat evaporates from that surface, it carries thermal energy with it and keeps the core temperature in range. The flush is not a symptom of effort. It is the cooling system doing its job, using the same network of vessels that carries oxygen to muscle to simultaneously manage body temperature during the effort those muscles are making. Every part of that response, the accelerating heart, the redirected blood flow, the flushed skin, the sustained delivery to working muscle, is the direct descendant of the cardiovascular innovations that began in Cambrian fish and were refined across 400 million years of pursuit and escape. The next time you feel your heart pound at the start of something physical, or feel your pulse in your throat after something frightening, pay attention to it for a moment. You are not just feeling your heart, you are feeling the part of you that was shaped by every chase and every escape in the evolutionary history of your lineage. You are feeling the delivery system that has been the difference between catching and being caught since the first vertebrates opened the first arms race in the history of complex life. That delivery system did not just make bodies faster, it made them more capable in every direction. And in doing so, it created a vulnerability that no animal carrying it could have anticipated. The river it built was fast. It was pressurized. It reached every tissue in the body. And roads that reach everywhere carry more than you intend. Before large animals built fast internal transport networks, an invader entering a multicellular body faced a different kind of world. It had to move through tissue cell by cell, spreading by direct contact or by secreting molecules that damaged the cells around it. The process was slow. It was containable. A localized infection could be surrounded and sometimes overwhelmed simply by the physical barrier of surrounding tissue. The body was a collection of neighborhoods, and a threat in one neighborhood did not automatically become a threat to every neighborhood at once. The closed circulatory system changed this completely. When life built a network of pressurized vessels reaching every tissue in the body, connected to a central pump circulating the entire volume of blood multiple times per minute, it did not build a delivery network for oxygen and nutrients alone. It built a highway running to every organ, every muscle, every corner of the body, continuously, without interruption, carrying whatever is in the blood to wherever the blood goes. A bacterium that reaches the bloodstream does not face a slow journey through resistant tissue. It faces a fast, pressurized ride to everywhere. This is not a hypothetical threat. Sepsis, the condition in which bacteria or their toxic byproducts enter the bloodstream and trigger a system-wide inflammatory response, remains one of the leading causes of death in hospitals around the world today. It is so dangerous not because the bacteria themselves are necessarily more lethal than those causing localized infections, but because the circulatory system delivers them and their effects throughout the body so rapidly that the damage becomes overwhelming before any targeted response can contain it. The same architecture that made large, fast animal bodies possible made a new category of catastrophic illness possible at the same time. Evolution did not solve this problem by abandoning the circulatory system. The benefits were too great and too fundamental. What it did instead was build a second system inside the first one, a system that used the same highways the pathogens used, traveled the same routes, and was present in the blood before any invader arrived. The immune system is, among other things, a circulatory solution to a circulatory problem. White blood cells are produced in the bone marrow and released into the bloodstream, where they move continuously through the body. They are not waiting in fixed positions for threats to come to them. They patrol. A neutrophil, one of the most numerous white blood cells in human blood, circulates through the entire body multiple times before it is replaced. It is not idle during that circulation. It samples its chemical environment constantly, reading the molecular signals dissolved in the plasma, checking for the specific patterns of chemistry that indicate damage or invasion. When a neutrophil detects those signals, something precise happens. The vessel wall near the site of infection changes its surface chemistry, displaying molecules that the neutrophil can grab onto. The neutrophil slows, rolls along the vessel wall, grips tighter, and then squeezes through the vessel wall itself into the surrounding tissue, moving toward the source of the signals by following a chemical gradient the blood has been carrying. The circulatory system has not just transported the immune cell to the neighborhood of the problem. It has provided the communication channel through which the problem announced itself and the road along which the response traveled. The molecules that coordinate this response are themselves carried in the blood. A local infection broadcasts its presence through the bloodstream. The immune response is coordinated through the bloodstream. Fever, one of the most ancient immune responses in vertebrate evolution, is generated when signaling molecules released at a site of infection reach the brain through the blood and instruct it to raise the body's temperature. The brain does not know where the infection is. It receives a chemical message that tells it an infection exists somewhere, and it responds in a way that affects the entire body simultaneously. In vertebrates, this kind of rapid whole-body coordination depends on the blood. Without a fast internal transport network like this, immune defense can exist, but it cannot be coordinated through the body in the same blood-borne way. The speed of the response, the ability to mobilize resources from distant sites, the capacity to raise body temperature everywhere at once, all of it flows from the same infrastructure that carries oxygen and glucose and heat. And then there is memory. When the immune system encounters a pathogen for the first time, the response is slow. It takes days to produce enough of the right antibodies, to expand the right population of immune cells, to mount an effective defense. In that window, the infection can cause significant damage, but some of the immune cells produced during that first encounter do not die when the infection is cleared. They persist in the blood and in lymphoid tissue for years, sometimes for a lifetime, as memory cells. If the same pathogen enters the body again, these memory cells recognize it within hours and mount a response so fast and targeted that the infection is often eliminated before it causes any symptoms. Your blood knows things about your medical history that your conscious memory does not. It carries cells shaped by infections you had as a child and may have no conscious recollection of. It holds the record of every vaccine you have ever received, encoded in populations of long-lived cells circulating quietly through your vessels right now, ready to respond to a threat that may never arrive. When you got your last flu shot, your arm was sore for a day and you thought nothing more of it. What actually happened was that your immune system spent the following 2 weeks building a library entry, manufacturing memory cells calibrated to a specific viral signature, and releasing them into the bloodstream, where they have been patrolling ever since. The river that life built to carry oxygen became a surveillance network, a communication system, a mobile army, a library of biological experience distributed through every tissue in the body without stopping, but carrying all of this, oxygen and immune cells and chemical signals and everything else, under pressure, through vessels running to every corner of the body, created a vulnerability that the earliest animals with closed circulatory systems had no way to anticipate. The system only works if the circuit stays intact. And circuits break. When they do, the pressure that makes everything else possible becomes the mechanism of destruction. The same force that drives oxygen to the farthest capillary in the body drives blood out of a wound with a speed and volume that can become fatal within minutes. Life had built the most powerful internal transport system in the history of animal evolution. Now, it had to figure out how to patch it. Consider what it means to run a fluid under pressure through a network of vessels that extends to every part of a living body. The vessels bend when the animal moves. They compress when a muscle contracts around them and expand when it relaxes. They twist when the body turns, absorb impact when it lands, flex with every breath. The body they run through is not a static container. It is a dynamic, continuously moving structure that subjects every vessel in it to mechanical stress thousands of times per day, every day, for the entire life of the animal. And the body moves through a world that damages it. Rocks and thorns and teeth and claws and the hard edges of everything the animal encounters in a lifetime of living in an environment that does not care about its survival. Every cut, every puncture, every abrasion that breaks the skin also breaks vessels. The pressurized fluid inside those vessels does not wait. It follows the physics of pressure and moves toward the breach, out of the circuit, away from the body, carrying with it the oxygen and the immune cells and everything else the animal depends on. This is the patching problem. A closed circulatory system running at high pressure has a failure mode that open systems do not face in the same way. If the circuit breaks, the pressure that makes the system so powerful becomes the mechanism of its own destruction. The same force that drives oxygen to the farthest capillary in the body drives blood out of a wound with a speed and volume that can become fatal within minutes. Evolution had to solve a problem with no elegant solution. It had to build into the blood itself a system that could detect a breach, seal it quickly, and do so without accidentally sealing vessels that were not breached. The system had to be fast enough to prevent catastrophic loss before anything else could respond. It had to be precisely localized, activating only at the site of injury and not throughout the circulation. And it had to be self-limiting, stopping when the breach was sealed, rather than continuing until the entire vascular network was blocked. What evolution produced is called the coagulation cascade. And it is one of the most finely balanced systems in vertebrate biology. The cascade is a chain reaction. It begins with damage. When a blood vessel wall is breached, proteins normally kept separate by the intact vessel wall come into contact with each other. This contact triggers the first step in a sequence of reactions in which one protein activates the next, which activates the next, each step amplifying the signal so that a tiny initial trigger produces a large and rapid response. The end product is a mesh of fibrin strands woven across the breach, trapping red blood cells and platelets and forming a clot that physically seals the opening. The amplification built into the cascade is extraordinary. A single molecule of the initial trigger enzyme can activate hundreds of molecules of the next enzyme in the chain, each of which can activate hundreds more. The signal grows at each step, which is why a clot can form within seconds of injury, despite the fact that the triggering event involves only a tiny number of molecules at the initial breach. Platelets contribute to this process in a way that is distinct from the protein cascade. When they encounter the exposed proteins of a damaged vessel wall, they change shape, extending projections that help them stick to the damaged surface and to each other, forming a temporary plug at the breach before the fibrin mesh has fully assembled. The platelet plug provides immediate mechanical closure. The fibrin clot that forms on top of it provides durable structural reinforcement. The precision required to make this work without causing clotting throughout the entire circulation is almost difficult to describe. Every step in the cascade is regulated by inhibitory proteins that prevent the reaction from spreading beyond the site of injury. The same blood that carries the clotting factors also carries molecules whose sole function is to neutralize those clotting factors the moment they drift away from the breach. The coagulation cascade is not simply switched on by injury and then left to run. It is continuously being switched off everywhere, except exactly where it needs to be active. Few systems in engineering have to balance two demands. This perfectly flow everywhere. Clot only where damage has occurred. The blood manages this balance in your body right now, constantly, without your awareness, adjusting in real time to the state of every vessel in the network. When this balance fails in either direction, the consequences are severe. Hemophilia, the inherited condition in which one or more clotting factors are absent or dysfunctional, removes a critical step from the cascade. A person with severe hemophilia A lacks functional factor VIII, one of the proteins in the middle of the chain. Without it, the amplification breaks down and the clot either forms very slowly or fails to form adequately. A cut that a person with normal clotting would seal within minutes can bleed for hours. Internal bleeding into joints and muscles, triggered by minor trauma or even by ordinary movement, accumulates over time and causes progressive damage. Before the development of factor replacement therapy in the 20th century, severe hemophilia was often fatal in childhood or early adulthood. The opposite failure is equally instructive. Deep vein thrombosis, pulmonary embolism, ischemic stroke, and myocardial infarction are all conditions in which clotting occurs where it should not, inside intact vessels rather than at a breach. A clot forming inside a coronary artery blocks the blood supply to the heart muscle it serves. That muscle, deprived of oxygen, begins to die within minutes. A clot in a cerebral artery produces the same result in neural tissue. These are not failures of the clotting system in the sense of it being absent. They are failures of regulation. Situations in which the careful balance tips toward clot formation inside the vessel itself. The same system, the same chemistry. One set of failures causes too little clotting, and the animal bleeds out. Another set causes too much clotting, and the animal clots from the inside. The system walks a narrow line between two forms of catastrophe at every moment of every day. This is not a flaw waiting to be fixed. It is what selection produced from the variation that existed. And what selection produced is a system that works well enough, most of the time, for most individuals, to allow reproduction and survival across hundreds of millions of years of animal evolution. Evolution is not interested in perfection. It is interested in good enough, often enough, to keep the lineage going. The coagulation cascade solved the patching problem well enough for the vertebrate lineage to survive and diversify across 400 million years. It kept the circuit intact through every injury that the animals carrying it survived long enough to reproduce from. And it carries within it a lesson that runs through every chapter of this story. The coagulation cascade exists because of the pressure. The pressure exists because of the closed circuit. The closed circuit exists because diffusion was too slow. The whole system is a chain of solutions to problems that earlier solutions created. Every answer generated the conditions that required the next answer. The pressure that made closed circulation so powerful also made every breach in the circuit potentially fatal. The coagulation cascade fixed the breach problem, but created a new failure mode in the wrong direction. Clotting where it should not. Each solution moved the lineage forward and left it standing in front of a new problem. This is the character of evolution. Not a march toward perfection. A continuous negotiation between what works and what the working creates. The coagulation cascade protected something that was becoming in the lineages that would eventually produce warm-blooded animals, increasingly worth protecting. A body that could afford to stay warm. A body that never stopped running. A body that, with enough blood arriving at the right rate, at the right time, could eventually sustain something that no cold-blooded body, with all its strengths, had ever been able to afford. A large and expensive and extraordinary brain. Somewhere in the Triassic period, roughly 230 million years ago, a small group of reptile-like animals began doing something that no vertebrate had done before. They kept themselves warm. Not warm the way a lizard is warm when it sits on a sun-heated rock in the afternoon. That kind of warmth is borrowed from the environment, absorbed from an external source and lost again when the source is removed. The warmth these animals maintained came from inside, generated by their own metabolism and held at a constant level, regardless of the temperature of the air or ground around them. This was one of the most consequential physiological shifts in the history of vertebrate evolution, and it was built entirely on the circulatory system. A cold-blooded animal, or ectotherm, regulates its body temperature by behavior. It moves into sun to warm up and into shade to cool down. When the environment is cold, its body temperature drops. Its metabolic rate slows. Its muscles become sluggish, and its nervous system processes information more slowly. A cold lizard is not a sick lizard. It is a lizard whose biochemistry is running at whatever rate the ambient temperature permits. This is not a bad system. Ectotherms are extraordinarily successful and have been for hundreds of millions of years. But the system has one constraint built into it that no amount of evolutionary refinement can remove. The animal's performance is bounded by its environment. It cannot be fast when the environment is cold. It cannot sustain high activity through a cold night or a cold season without paying the price of reduced capacity. The warm-blooded animals, or endotherms, escaped much of this constraint by generating their own heat through metabolic activity and distributing that heat throughout the body via the blood. The circulatory system is central to this in two ways. First, it is the mechanism of heat distribution. Warm blood generated by metabolically active core organs, the liver, the heart, the muscles, flows outward through the arterial system to every part of the body, carrying thermal energy along with oxygen and glucose. A warm-blooded animal maintains a consistent temperature in its extremities, its brain, its sense organs, because the blood arriving there is warm, regardless of what the environment outside is doing. The body is not at the mercy of the thermometer outside. It carries its own thermometer and maintains its own set point. Second, the circulatory system is the mechanism of heat management. Generating internal heat is energetically expensive, and the body must be able to shed excess heat to avoid overheating during intense activity. Blood vessels near the skin surface dilate during exertion, routing more blood close to the surface, where heat can radiate away and where sweat evaporation can carry thermal energy off. When the body is cold and needs to conserve heat, those vessels constrict, keeping warm blood circulating near the core. The skin's blood supply is a continuously adjustable thermal radiator controlled by the same autonomic nervous system that adjusts heart rate and vessel diameter throughout the body. You feel this every time you exercise hard enough to flush. The redness in your face is not a sign of distress. It is the blood routing itself toward the surface to shed the heat your muscles are generating. The system is doing exactly what it was built to do, using the same network that carries oxygen to muscle to simultaneously manage the temperature at which those muscles are working. The cost of all this is enormous. A warm-blooded animal at rest burns roughly 5 to 10 times more calories per unit of body mass than a cold-blooded animal of comparable size at the same temperature. A mouse and a lizard of the same weight sitting still in the same room are not metabolically comparable. The mouse is burning through its energy reserves at a rate that would exhaust the lizard's equivalent reserves in a fraction of the time. Small, warm-blooded animals must eat almost constantly. A shrew must consume close to its own body weight in food every day, simply to sustain its metabolic rate. It cannot miss a meal the way a snake can go weeks between feedings. What the body gets in return for that fuel expenditure is independence. Independence from the temperature of the environment. Independence from the time of day, the season, the altitude, the latitude. A warm-blooded animal can be active, alert, and at full physical capacity on a cold night, in a cold winter, at a high elevation, in ways that a cold-blooded animal of comparable size simply cannot match. This independence opened ecological niches that had been inaccessible to vertebrates, and it created the foundation for a further development that would eventually change the planet more than anything that had come before. The brain. Not all brains. Not the early, compact brains of fish and reptiles, which are remarkable organs in their own right, but which operate within metabolic budgets their bodies can sustain without dedicated high-volume blood supply. The large, metabolically expensive brains of birds and mammals. And above all, the extraordinarily large brain that appeared in the human lineage. The human brain accounts for roughly 2% of the body's mass. It consumes roughly 20% of the body's resting energy budget. This ratio, 10 times what its mass alone would predict, reflects the metabolic intensity of neural tissue. Neurons maintain electrical gradients across their membranes continuously, pump ions against those gradients continuously, synthesize neurotransmitters continuously, and communicate with thousands of other neurons continuously. all of which requires a constant and uninterrupted supply of oxygen and glucose. The blood supply to the brain reflects this demand with corresponding precision. The brain receives blood through major arteries that merge into a connected ring at the base called the Circle of Willis, an arrangement that provides redundant pathways so that the blockage of a single vessel does not necessarily cut off entire brain regions. From this ring, smaller arteries branch into the brain tissue, subdividing into capillaries so densely packed that no neuron in the cerebral cortex is more than about 25 micrometers from the nearest capillary. 25 micrometers, well within the range where diffusion can cover the final step. The entire architecture of the brain's blood supply is built to ensure that the metabolic demands of neural tissue are met with a reliability that no other organ requires to the same degree. When that supply is interrupted, the consequences are immediate in a way that reveals how completely the brain depends on continuous delivery. A cardiac arrest that stops blood flow to the brain produces loss of consciousness within seconds. Brain cells in the most metabolically active regions can begin to sustain damage within minutes of oxygen deprivation. Consciousness is not made of blood, but without blood arriving on time, consciousness disappears. The most complex, most expensive, most capable structure that evolution has ever produced cannot continue without the most consequential fluid that evolution has ever produced, delivering what it needs, without interruption, every moment of every day. The thought you are having right now depends on a pump that began working before you ever took your first breath. It depends on vessels that have been delivering oxygen to your neural tissue without interruption since before you were born. It depends on hemoglobin molecules loading and unloading iron-bound oxygen in capillaries thinner than a hair. Diffusion, covering the last 25 micrometers. Neurons firing in patterns of extraordinary complexity on the energy that delivery provides. The warm-blooded brain is the most expensive thing the circulatory system has ever been asked to support. It is also, by any measure that the organism carrying it can apply, the most valuable, and it is the thing that made it possible, eventually, for a lineage of primates to sit in a world built on blood and begin to ask where blood came from. That question is what the last chapter of this story is built around. Not what blood does, not how it works, not what happens when it fails, what a single drop of it contains when you look at it, knowing everything that had to happen to put it there. Take a drop of blood, a single drop, small enough to balance on the tip of a finger, so ordinary that most people spend their lives never giving it a second thought. Put it under a microscope. What you are looking at is not a simple fluid. It is not merely a solution of chemicals suspended in water, though it is also that. What you are looking at is 600 million years of solved problems, compressed into a volume smaller than a raindrop, still working, still running every function it was shaped across that unimaginable stretch of time to run, in the body of an animal that can now look at it and ask what it is. The red cells are the most immediately visible. In a single drop of human blood, there are roughly 5 million of them per cubic millimeter, so densely packed that they give blood its color. Each one is a biconcave disc, shaped like a pillow pressed from both sides, flexible enough to bend through capillaries narrower than its own diameter without tearing. There are no nuclei in these cells, no DNA, no capacity to divide or repair themselves. The human red blood cell has been stripped down to its essential function with a ruthlessness that evolution reserves for things that need to be done with absolute efficiency. It is a vessel for hemoglobin and nothing else. Performing that function for roughly 120 days before it is broken down and its components recycled. The iron extracted and routed back into the bone marrow to be incorporated into new hemoglobin in new red cells. The iron in those cells is old in a way that is genuinely difficult to sit with. Iron has been moving through living chemistry for billions of years, passing from organism to organism, from generation to generation, cycling through the ocean and the soil and the blood of countless creatures across the entire span of animal evolution. The iron atoms sitting at the center of your hemoglobin were forged in the collapse of a massive star before the solar system existed. They have been part of living things since before the first fish opened the first arms race in a Cambrian ocean. They are in you now. You are their current address. Look past the red cells at the plasma, the pale yellow liquid in which everything floats. The plasma is roughly 90% water, and that water carries in solution a collection of molecules that reads like a history of every problem animal bodies have ever had to solve simultaneously. Albumin, the most abundant plasma protein, is a transport molecule of extraordinary versatility, carrying fatty acids and hormones and calcium and dozens of other molecules that need to travel through the bloodstream without dissolving into the vessel walls or triggering immune responses. It is also the molecule responsible for maintaining the osmotic pressure that keeps fluid inside the vessels rather than leaking into surrounding tissue. Without albumin, blood plasma would seep through the walls of capillaries and accumulate in tissue, and the vessels would gradually empty. Albumin does not carry oxygen or fight infection. It is the molecule that keeps the river in its banks, and it has been doing that job across the entire history of the vertebrate lineage. The glucose dissolved in the plasma is the immediate fuel that every cell in the body runs on. Its concentration in the blood is maintained within a remarkably narrow range by an interlocking system of hormones, primarily insulin and glucagon, that respond within minutes to rises or falls in blood sugar. Too much glucose and the osmotic properties of the blood change in ways that damage vessel walls over years. Too little, and the brain, which cannot store its own glucose and cannot function on alternative fuels the way muscle can, begins to fail within minutes. The glucose in your blood right now is being held at a concentration that your cells evolved to expect, maintained by a regulatory system refined across the entire span of mammalian evolution. And it is being delivered continuously to every cell in your body by the same circulatory system that carries everything else. The clotting factors, dissolved invisibly in the plasma, are the entire coagulation cascade held in reserve, ready to activate within seconds of vessel damage, carrying the solution to the patching problem that evolution spent hundreds of millions of years developing. They are present at all times in every cubic millimeter of blood, circulating quietly, kept inactivated by inhibitory proteins that prevent them from triggering the cascade spontaneously, waiting for the specific signal of vessel wall damage that will set the chain reaction in motion. Now look at the white cells. They are far fewer in number than the red cells. Roughly one white cell for every 500 to 1,000 red cells. But what they lack in numbers, they compensate in complexity. A neutrophil circulating past in the plasma carries within it the ability to detect bacterial proteins, squeeze through vessel walls, track chemical gradients through tissue, engulf and destroy pathogens, and release signals that coordinate the response of other immune cells. It learned to do all of this, not in your lifetime, but across 400 million years of vertebrate immune system evolution. Each refinement selected because it improved the odds of surviving infection long enough to reproduce. The lymphocytes carry something even more remarkable. Some of them are memory cells. shaped years or decades ago by a specific infection or vaccination, carrying on their surface receptors, calibrated to recognize a specific pathogen with a precision that took the immune system days to develop during the original encounter. They have been circulating ever since. Patient, long-lived, maintaining in their molecular structure the record of a threat that the body survived and that the blood now stands ready to recognize again within hours rather than days. Your blood carries a library of your immunological biography. Every significant infection you have survived. Every vaccine you have received. Every pathogen your immune system learned to recognize and defeat has left its record in populations of memory cells still moving through your vessels. The blood does not just transport, it remembers. And the memory it holds is not symbolic or metaphorical. It is molecular, specific, and functional. It is ready to act on what it knows the moment the relevant signal appears. Blood still carries echoes of the watery, mineral world in which animal cells first learned to live. The salt concentration of blood plasma, the balance of sodium and potassium and calcium and magnesium dissolved in it, reflects a chemistry that was established when the ancestors of vertebrate cells were surrounded by the ancient ocean. As animal life moved onto land, the body did not abandon that internal chemistry. It enclosed it. It maintained the conditions that the cells had evolved to require, even when the external environment no longer provided them. The salt in your blood is not a preserved sample of an ancient sea. It is an inherited chemical echo of the world that animal cells never fully left behind. When you taste blood from a cut and notice something faintly like salt water, you are tasting a chemistry that your cells have been maintaining for hundreds of millions of years, long after the ocean that first shaped it receded from the evolutionary story of your lineage. Now, consider what all of this together actually represents. The red cells with their ancient iron, the plasma with its evolved chemistry, the clotting factors holding the patching solution in reserve, the immune cells carrying the record of past encounters, the glucose held at the concentration that evolution set as the standard for vertebrate cellular function, all of it moving continuously, driven by a pump that has not stopped since before you took your first breath, through 60,000 miles of vessels calibrated to deliver the right things to the right places at the right rates, in response to what the body is doing at this exact moment. A drop of your blood is not just a fluid. It is a museum that is still working. The other museums you have visited hold objects from the past in cases behind glass, preserved and inert, separated from the world that made them by centuries or millennia. The museum in your blood holds objects from a past measured in hundreds of millions of years, and every one of them is active, functional, and doing right now exactly what it was shaped across that unimaginable span of time to do. The hemoglobin loading oxygen in your pulmonary capillaries is running the same molecular mechanism that the first hemoglobin-carrying vertebrates ran in a Silurian ocean more than 400 million years ago. The coagulation cascade, ready to activate at any vessel breach, is built on proteins that were already present in the ancestors of jawed fish. The memory lymphocytes, carrying the record of your past infections, are the latest iteration of an immunological memory system that has been running in one form or another since before the first land vertebrates crawled out of shallow water. None of this was designed. None of it was planned. It accumulated, piece by piece, through the same blind process that built everything else in the living world: variation, selection, and time. The variation was random. The selection was indifferent. The time was almost incomprehensibly long. And the result is a fluid of such functional complexity that researchers are still discovering things about it, still finding molecules in it whose roles were not previously known, still uncovering regulatory mechanisms whose existence had not been suspected. You are not simply carrying blood. You are, in a deeper sense, the current expression of everything that blood made possible. The size of your body, well beyond the few millimeters that diffusion would allow without it. The speed at which your muscles can contract and sustain contraction. The stability of your body temperature across the range of environments your species has learned to inhabit. The capacity of your immune system to recognize and remember threats. The metabolic intensity of the brain you are using right now to follow this thought from one sentence to the next. All of it runs on the river. The river that life built when the wall of diffusion became a ceiling it could not afford to stay under. The river that carried iron from dead stars to every cell that needed it. The river that made the first chase possible, and the first sustained flight, and the first warm night spent alert and capable when every cold-blooded competitor was slow. The river that became a road for invaders and then became an army to patrol that road. The river that learned to patch itself and to remember its enemies and to distribute heat to a brain large enough to wonder where rivers come from. That river is still moving. Right now. In you. The museum is open. Go back to the beginning of this story. A world without blood. A world where the most complex living things were pressed flat against the seafloor, spread thin across rock, shaped by the unyielding arithmetic of a physical law that said oxygen could drift only so far before it ran out. The wall was real. It set a hard limit on what life could build. And for hundreds of millions of years, everything alive stayed on the right side of it. The physics of that wall has not changed. Not once in 600 million years. Oxygen still moves through biological tissue at the same rate it always has. The 200 micrometer limit is as absolute today as it was in the Ediacaran Ocean. The wall is still there. What changed was not the wall. What changed was what life learned to carry to the foot of it. The ocean outside is where life began. The ocean inside is what let life leave. That sentence appeared near the beginning of this story as a promise. It has a different weight now. It is no longer a description of what blood is. It is a precise account of what happened. Life began in water, surrounded by the chemistry that cells evolved to require. When it built bodies too large and too complex to stay in contact with that external water everywhere at once, it solved the problem by enclosing a version of that water inside itself. It maintained the salt concentration, the mineral balance, the chemical environment that animal cells could not survive without. And it carried that environment to every cell, regardless of what was happening outside the body. And then it did something more. It pressurized that internal ocean. It loaded it with a molecule built around ancient iron that could carry oxygen far beyond what plain water could manage. It built a pump of extraordinary endurance to drive it through a network of vessels, calibrated to deliver the right cargo to the right place at the right moment. It armed that ocean with a surveillance and defense system that used the same network to patrol for threats and remember every significant enemy the body had ever faced. It built into it a self-sealing mechanism that could close a breach in the pressurized circuit within seconds. And it used the whole system to maintain a body temperature independent of the environment, which freed a brain from the constraint that had limited every cold-blooded animal before it. The river is ancient. What it is currently feeding is very new. The circulatory system has been running in one form or another for 600 million years. The lineage that became human has existed for perhaps 300,000 years. The capacity for the kind of historically aware thought that lets a person trace blood back through Cambrian oceans and dead stars, and the first arms race in the history of complex life has been around for far less than that. The river predates the brain it is currently supplying by an almost incomprehensible margin. And yet here you are, aware, in some way, of both the antiquity of the system running through you and the novelty of the awareness itself. You did not earn the blood. You did not build the hemoglobin or design the coagulation cascade or evolve the four-chambered heart. You inherited all of it. The way every animal that has ever lived inherited the solutions that the lineages before it developed. You are the current recipient of 600 million years of accumulated answers to questions that the living world never asked consciously, but answered anyway. Through the slow, indifferent mechanism of what survived and what did not. The blood does not need you to know what it is. It has been doing its work since long before anything alive could know anything at all. But you do know, or you know more than you did when this story began, which may be the only kind of knowing that is ever fully available to something as new as a conscious mind, trying to understand something as old as the fluid keeping it alive. The wall is still there. The ocean you carry got you past it. And right now, without your awareness, without your instruction, without asking your permission, it is still moving.